Hi everyone, hello. How are you doing? I hope you are doing good. So welcome to another video of chapter 9, which is control and coordination. We were talking about the structure of the neuron. Then we went on discussing the types of neurons and the types of neuroglial cells which are surrounded, which are present in the surrounding of the neurons. They help the neuron in their proper functioning, like oligodendrocytes, ependymal cells, Schwann cells, then astrocytes, macrophages, microglial cells. Okay, all this we have discussed up till now. Now we will move to the properties of the nerve fibers. Having understood the structure of the nerve fibers and the surrounding cells of the nerve fibers, now we will discuss what are these nerve fibers characteristics, what do they do, what are their characteristics, what are their properties. Then we will discuss the types of synapses. We are going to discuss in more detail about the chemical synapses. Okay, because in humans mostly the type of synapse which is present is chemical. Okay, let's begin. Properties of the nerve fibers. Before understanding the properties, we will just go through the synapses again. We will revise. What is a synapse? You have neuron 1 and then you have neuron 2. The axon terminal of neuron 1 and the dendron, the beginning. Okay, the dendron, the beginning of the another neuron. This is 1, this is 2. So the space between the terminal of a neuron and another neuron or a muscle or a gland. So this can be either a muscle or a gland or a neuron. Okay, so the space which is present in between the junction, this is known as the synapses. The impulse has to be traveled from here to here via this space, this junction or this synapse. Okay, so before understanding the types, let's just discuss the properties of the nerve fibers. Number one is excitability. What do we mean by excitability is that the capability of the neuron, the capability of the nerve fiber to carry, to conduct the electrical impulse. Okay, this is because of the polarized membrane that is present. Okay, what is polarization? We will be seeing in the further videos when we will discuss the mechanism of the electrical impulse transmission. There will be we will be seeing how this electrical impulse is transferred to the membrane of the neurons. So right now, just the property, that excitability, that is the nerve fibers have the capability of transmission, of conduction of this electrical impulse because their membrane can be polarized and repolarized. Okay, then next is conductivity. That is the ability of the neuron to transmit. It's just not sitting with the information. It is capable of passing on the information. Okay, capability of transmitting the information, the excitation signal. Summation effect is that for a nerve fiber or for a neuron to to carry the impulse or to transmit the impulse, okay, to get excited, it requires a minimum threshold value of that particular impulse. That impulse can be electrical, chemical, or physical, anything. So that detectability of the impulse should be of certain range, should be of certain value, the threshold stimulus we call it as. Okay, so that minimum threshold level should be present for the neuron to get excited. That is known as a summation effect. Okay, sorry. So that is the threshold effect. So if there is a signal below the threshold value or below the threshold value, then the neuron will not be excited. The neuron will not be excited. If it is more than the threshold value, then the neuron will be excited at the normal level for which, the normal threshold stimulus or the threshold value, which is minimal required. So it will excite to that certain only. This does not mean if the signal is more, if the excitability, the threshold value of the coming information or coming electrical impulse is more, so the excitability will be more. No, the excitability will be normal, even if the signal that is coming has the threshold value, minimal threshold value. But if it is low, then the neuron will not be excited. So summation effects tells what? If there are any weak signals which is below the threshold value, okay, If there is only one weak signal that is coming below the threshold value, then it will not excite the neuron. But if there are multiple weak signals coming one after the another immediately, which are below the threshold value, then they will be capable of exciting the neuron. So that is what is meant by summation effect. So multiple weak signal can come and together they are able to excite the neuron. Then another property of the neuron is that all or none. This means what? A nerve fiber or a neuron will conduct the electrical impulse, okay, through the entire length of the axon. Yes, either it will carry throughout the axon or it will not carry the signal at all. Yes, so it is not like half the part of the neuron might be involved in electrical impulse conduction or transmission, half is not involved. Like this does not happens. Okay, so the nerve impulse will either conduct the impulse along the entire length or it will not conduct at all the impulse. Okay. Then next is refractive period. What is this refractive period? This is the period wherein the nerve fibers fails to any strong impulse that is coming. So if there is a strong signal, strong electrical impulse that is coming, there will be certain fraction of seconds for that period, the nerve will not conduct anything. It will fail to receive that signal. That is known as refractive period. Then in line with the refractive period, you have synaptic delay. This synaptic delay is for mostly for a few milliseconds, you can say. Okay, this is basically when the neurotransmitters, the chemical neurotransmitters are getting released. So the time that is required for the neurotransmitters to be released, it is approximately 0.3 to 0.5 milliseconds. So that time period, that delay which is there, by the time the neurotransmitters are released and then the signal is transferred to the next neuron, so that is known as synaptic delay. This happens at the junction, at the synapses between the two neurons. And then finally you have the synaptic fatigue. That is when the neuron is involved in a single transmission, there will be a time wherein it will just sort of exhaust itself. It will sort of, stop single transmission. It will halt for a few milliseconds. Okay, it will halt or it will pause and then it will continue the conduction and transmission of the electrical impulse. And finally is the velocity, that is the speed at which the impulse is transmitted. Okay, so these are the properties of the nerve fibers: excitability, conductivity, stimulus and summation effect, all or none effect, refractive period, synaptic delay, and fatigue, and finally the velocity. Moving to the chemical synapses. Very simple to understand. There can be two types of synapses, electrical or chemical. Okay, we will be focusing more on chemical. Okay, we will be discussing the difference also simultaneously. In this picture, you see this is the terminal of one neuron and this is the another neuron. What is that name? Now we will be calling it as that is the preceding neuron, presynaptic neuron, and this is the the next neuron, the postsynaptic neuron. In case of electrical, there is very little space between the two. So the synapse is very, very, very close together, And you have this special gate, you can say, special gate or opening, which are known as gap junctions. Okay, there are different types of these communication channels, you can say. You have gap junctions, you have intermediate junctions. Okay, then in plants you have plasmodesmata. So these serve as communication channels. So when there is an electrical synapse, so gap junctions will be present, and the presynaptic neuron and the postsynaptic neuron, they are very close together. Okay, and directly the electrical signal will be transferred from the pre to the post directly through these gap junction. Okay, so we will see here electrical transmission occurs at an electrical synapse, chemical will occur at a chemical synapse. In electrical case, the transmission is faster. In chemical, it is slower, because here you see in chemical synapse, there are these vesicles, these vessels which carry the chemicals which will be released, and then only the signal will be transferred to the next membrane, and then the space is more. As you see, the space between the presynaptic and the prosynaptic neuron is much bigger as compared to the electrical synapse. Okay, so the vesicle has to be made, it has to be released, it has to be fused with the postsynaptic membrane, and then only it will go further and cause the excitation of the following neuron. So in case of electrical transmission, the electric current transfers directly from one neuron across the synapse to the another neuron. Whereas in chemical neurotransmitters, the chemical are involved in the transmission of the impulses at the synapse. So here there is direct communication. Here there is middleman. What is the middleman over here? Here the middleman is the chemical messengers, or you can say neurotransmitters. Fine. Here, in case of electrical, the membranes, the pre and the postsynaptic membrane neurons are very close proximity, are in very close proximity. Here, they are separated by a cleft. There is a space. This is known as cleft. Okay. Electrical transmission is very rare in our system, and chemical transmission is most common in our system. And that is why we are going to discuss the chemical synapse in a bit detail now. Okay, so this is the difference between the chemical synapse and the electrical synapse. Now moving forward to the chemical synapse. So this is how a chemical synapse will look like. Again, we will just go through the labelings. This is the neuron. Okay, this is the pre-synaptic neuron, and this is the post one. Okay. Yeah, this is the axon terminal of the previous neuron, and this is the dendrite spine of the next neuron. You have these channels again. These are not gap junctions. These are receptors. This is the receptor on the postsynaptic neuron. These are channels, or you can say gate. Okay, they will open and close. Now you can see the labeling: voltage gated calcium channels. Means what? This channel, this gate will open when there is calcium ions over here. It will be closed when there is no calcium ion over here. So hence the name. This channel is only opening when there is calcium ions. In absence of calcium ion, they will remain closed. And these are the vessels which will carry the chemical information or the neurotransmitters, as you say. The signal is transmitted in form of these neurotransmitters. Okay, so they are known as synaptic vesicles. So let's begin. How does an electrical transmission or impulse takes place via this chemical synapse? So the first step is, this is your presynaptic neuron. This is a neuron. And when the axon terminal of this neuron, this is one neuron, and then the cell body, the dendrite of this another neuron. So when you magnify this, this is how the picture looks like. Okay, so this is your presynaptic neuron. Step number one, signal is coming. Action potential arrives. What is another name for the signal? Action potential. Okay, it is coming now. It arrives at the axon terminal. Okay, now as soon as the signal is received, what will happen? What will happen? Calcium ions will enter over here, and they will open the channels. Voltage gated calcium ions channels will open, and calcium enters the axon terminal. So here you will see calcium ions. As soon as the action potential or the signal comes, calcium ions will come in and they will open the gate. As soon as the gate is open, calcium entry causes these neurotransmitters containing vesicles to release their substances, to release their contents by exocytosis. As you see in number 3, once the calcium ions are open, these vesicles will now migrate. They will come here, they will fuse with the membrane, and they will release their neurotransmitters. These dot things you see in the presynaptic axon terminal, they were contained in a vessel. So once it opens the gate, the vessel will give away all their neurotransmitters. And then this neurotransmitters, which is there in this space, in this synapse, which is the synaptic cleft, they will now diffuse with the following neuron. You will see receptors over here. These are the receptors, okay, to which the neurotransmitters will bind, okay, bind to this ligand-gated ion channels, and then the information will be transferred forward. How it will be forward and how the there is change in the polarity of the membrane of the neuronal membrane, that we will see in the next video when we will actually see the conduction of and the transmission of the electrical impulse from one neuron to the another. Right now, we are just understanding the junction, the chemical synapse. What happens at the chemical synapse? Okay, let's just go through this again. So step number 1, signal is coming, action potential arrives at the axon terminal. Step number 2, so step number 1, signal is coming. Step number 2, channels will open, calcium ions will enter, okay, and increase in the calcium ions will cause what? Calcium ions will signal these synaptic vesicles to go and now fuse with the membrane. Okay, vesicles will move towards the membrane, and then they will release the neurotransmitter by exocytosis, okay, by giving away the neurotransmitters. Then the neurotransmitters are now in the synaptic cleft. Number 7, they will bind to the receptor. Okay, they will bind to the receptor, and then further the information will be taken forward. Now that how it is taken forward, that we will see in the next video. Okay, so this is how what a chemical synapse looks like, and what are the events in the transmission of the neurotransmitter from presynaptic axon to the postsynaptic neurons. Okay, so that's it. Now in the next video, as I am talking that transmission of the nerve impulse, we will cover it in the next video, and how this nerve impulse is generated and conducted. So keep revising, make a note of your doubts, and reach out to me in case of difficulty. Thank you. Take care. See you in the next video. Bye.